Hey guys, Miss Cote here, and welcome to this episode on gene regulation. In our last video, we looked at how bacteria, otherwise known as prokaryotes, can turn on or turn off genes by organizing them into complexes called operons. In this episode, we'll look at a variety of ways that our cells, otherwise known as eukaryotic cells, can control whether or not we transcribe and translate certain genes. So let's get started. Think about this for a minute. You have trillions of cells in your body, and they can be any one of 200 different cell types, specialized to perform particular jobs to help you run smoothly. But even though we have a wide variety of cell types, all of them have the same DNA housed in their nuclei. So every cell in your body has all of the instructions to make any polypeptide that our body may need. But just because we have the instructions to make a protein, that doesn't mean that we have to make it. Specific cell types only need to make the proteins that they need to function. This could only be a small percentage of the total number of genes that it could use. For example, a muscle cell only needs to make certain proteins for it to work properly, and those proteins may be different than what the cells in your eyes need to make. This is called differential gene expression. But this is not only turning on specific genes, but also making sure they're turned on or turned off at the right times. Otherwise, this could lead to issues harmful to the organism. So in this unit, we've been looking at the processes involved in gene expression, transcription, RNA processing, and translation. And with any pathway, there can be control points to stop or stimulate the process. This diagram in your book is a fabulous overview of the many control points involved in regulating gene expression. If you're into sketch notes, this would be the diagram to base your notes off of for this topic. I'm going to keep referring to this diagram as we move through the different control points. First, let's take a look at the structure of chromatin itself. Remember that chromatin is composed of DNA and proteins. And those proteins are called histones, in case that ever comes up. This is the form your genetic material is in when the cell is not dividing. Chromatin can be packed tight or loose, and that can play a huge role in whether or not the gene you want is actually accessible. We can chemically modify the chromatin to make it tighter or looser, depending on what the cell's needs are. Let's take a look. The first way we can influence chromatin is by modifying the histones, the proteins. Adding acetyl groups to amino acids, specifically lysine, will loosen the structure, making it easier to start transcription, while adding methyl groups to amino acids will tighten up the structure, therefore making it harder to access the gene. Here's a diagram of histone acetylation. I know we really didn't go into chromatin packing, but DNA, shown in blue, gets wrapped around a complex of 8 histones, shown in purple. This structure is referred to as a nucleosome and looks like beads on a string. Nucleosomes can be packed even tighter, which eventually leads to the condensed chromosome structure for cell division. On the bottom of the slide, it's showing that the addition of acetyl groups to the tails of the histones will cause the chromatin to spread out and loosen up. And while there's no diagram showing methylation, it acts in the opposite direction and tightens up the chromatin. The other type of chromatin modification would be to modify the DNA itself. This could be done by adding methyl groups to bases, more specifically cytosines. Methylation inactivates or silences the gene and it will not be expressed. Methylation is why we have genomic imprinting. Remember back in genetics, we saw how the phenotype can depend on which parent you got your alleles from. Scientists who study genomic imprinting have found that the inactive genes are due to heavy methylation of the DNA. So we've just seen examples of altering gene expression without even mutating the DNA. Modifying the chromatin by sticking functional groups onto the chromatin can affect expression, and these modifications can be passed on to future generations. Epigenetics is an emerging field to study these changes in organisms that are caused by altering gene expression instead of the genetic code. Scientists are finding that what you eat, where you live, who you interact with, sleep, exercise, etc., can cause chemical modifications to your genes that can make them turn on or off. One of the best examples of epigenetics that seems to stick with you guys involves an interactive activity called Lick Your Rats. This activity takes you through how the ability of an adult rat to calm down after stress depends on how much mom paid attention to it during the first week of life. All rats have the gene. The level of nurturing will affect how much the gene is actually expressed. So we've just seen if a gene is made accessible at all by modifying the chromatin structure. If the gene is accessible, the next step would be to start transcription. If you remember, transcription is done in 3 steps: initiation, elongation, and termination. So if we want to regulate transcription, it would be to the cell's benefit to work at the initiation stage. So just to refresh our memories of the organization of a typical eukaryotic gene, you should already know that the gene itself is represented in blue with both exons and introns shown. At the beginning of every gene, there's a promoter region which includes the TATA box, shown here in green. This is the region that RNA polymerase will recognize and bind to start transcription. Also associated with most eukaryotic genes are segments of non-coding DNA called control elements. Here we have 2 regions of control elements. The one close to the gene is called the proximal control element, while the other one can be located far away upstream from the gene. This one's called the distal control element. The job of these control elements is to act as binding sites for regulatory proteins called transcription factors. Transcription factors can either activate or inhibit transcription. At the proximal control element, general transcription factors work for the transcription of all protein-coding genes. We saw this when we learned about the transcription initiation complex. Transcription factors bound to the promoter area to help RNA polymerase bind and start transcription. However, the distal control element is where specific transcription factors bind. Let's look at an example of how it works. Here we have a gene with a distal control element. Let's just pretend for the sake of space that this is way far upstream of the gene. The distal control element can also be referred to as an enhancer. In this example, the enhancer has 3 regions where activators can bind. And when activators bind to the enhancer, this sets off a series of protein interactions to stimulate transcription of a gene. Binding will bring in a DNA bending protein that will actually fold the DNA so that the enhancer, which remember is not neither gene, comes into close contact with the structural gene. And along with a complex of proteins, this will bring RNA polymerase to the promoter to start transcription. So while activators will stimulate transcription, please know that some transcription factors can act as repressors, which will inhibit the start of transcription. So the use of transcription factors is a method to regulate, either begin or inhibit, transcription. As I said in the beginning of this episode, certain cell types only need to make specific proteins. So how do cells make sure they're only making their proteins, and not the ones they don't need? This is done through a specific combination of activator proteins. Transcription of a protein will only happen if the cell has the correct combination of transcription factors. For this example, let's color code them. Here we have 2 different cell types, a liver cell and a lens cell. Obviously, these 2 cells have very different functions. The liver cell needs to make albumin, while the lens cell needs to make crystalline for the eye. So the activators found in the nucleus are only the ones to make the proteins that the cell needs. In order to make albumin, the red, gray, and yellow activators are required. And to make crystalline, gold, gray, and pink activators are needed. Notice that even though gray is common to both, it can't turn on the second gene just by itself. It needs the other 2 specific activators to turn on transcription. Using combinations of activators is also how related genes can be turned on at the same time. Remember, eukaryotic genes do not have operons, where all the genes in a related pathway are organized together. These genes could be spread out over the 23 pairs of chromosomes. But if all these related genes have the same control elements and activators, the genes can be transcribed at the same time to provide the cell with maximum efficiency. That takes care of ways transcription can be regulated. If transcription occurs, a piece of RNA is made. The next step would be to process the RNA into mature messenger RNA so that it could leave the nucleus and go into the cytoplasm for translation. So let's look at how RNA processing can be regulated. Remember that RNA processing takes place in 3 steps: the 5' cap, the 3' tail, and splicing the introns out and joining the exons. Splicing is the step that can be regulated. Regulation can happen through what's called alternative RNA splicing. One RNA transcript can produce different messenger RNA molecules, depending on which segments are treated as exons and which are treated as introns. Here in this example, one messenger RNA transcript is made up of exons 1, 2, 3, and 5, while the other is made up of exons 1, 2, 4, and 5, and these will each make different polypeptides. So at this point, let's assume that the mature messenger RNA has been made and has been transported into the cytoplasm for translation to occur. We can regulate transcription, and when we're finished with the messenger RNA, we can degrade it, break it down so that the nucleotides can be reused to build a different RNA molecule. Translation can be regulated in a couple of ways. Remember when we learned about translation in the last chapter? Translation began with the formation of a translation initiation complex. The messenger RNA, a transfer RNA, the ribosome, and some helper proteins need to all come together to get translation started. One way to block translation is to modify one or more of the helper proteins. So if the complex can't form properly, translation won't be able to happen. Another way to block translation is to have something bind to the messenger RNA so that it can't bind to the ribosome. This could be done by proteins or by small pieces of RNA, like microRNAs. Here's a diagram of how micro or miRNA works. These small pieces of RNA can bind to a complementary sequence on the messenger RNA. If the sequence is not completely complementary, it can just block translation. It just gets in the way. However, if it is completely complementary, it can trigger the degradation of the messenger RNA. The use of small non-coding pieces of RNA to do these things is also referred to as RNA interference, or RNAi. And last, once the polypeptide is made, regulation can happen through either allowing or not allowing the protein to fold properly, or by degrading the protein when it's no longer needed. Like messenger RNA degradation, the subunits, in this case amino acids, can be reused to build other proteins. Again, in order to be functional, the polypeptide needs to fold and sometimes be modified. Not allowing these things to occur can regulate whether or not the protein will do its job. And when a protein needs to be degraded, it's tagged or marked with a molecule called ubiquitin, and a complex called a proteasome will recognize it and break it down. Hence the meme on the intro slide. I know this was a lot of information to take in for one episode, so thanks for hanging in there. Just know that even though a cell has the capacity to carry out gene expression, transcription, and translation, there are methods to regulate these processes so the cell only needs to make what it needs. That way, the cell doesn't waste time, energy, and resources and is as efficient as it can be. So until I see you next, be a good human.